Good morning, afternoon, or evening, depending where you're joining from around the world today. And firstly, let me introduce myself. I'm Brian Brookman, and I'm the Chief Scientific Officer for Proficiency Testing at LGC. I'm very pleased to be able to chair our panel discussion today on optimizing cannabis testing in your lab, which is being hosted by Dr. Ehrenstorfer, LGC AXIO Proficiency Testing, and Digamma Consulting. In the last 10 years, we have seen the legalization of recreational and medical use of cannabis across the world. And this is a fast-moving topic, and associated with it is an increasing level of testing being carried out. And this is a complex area, with cannabis containing more than 500 unique compounds, including over 100 chemical alkaloids, known as cannabinoids, many of which have still not been fully characterized. And we've received a great number of questions already from many of our customers. So I'm really pleased to have a panel of experts with me today who will help tackle these questions. So let me just perhaps firstly introduce the panel. Firstly, I need to just make a quick apology. Unfortunately, Dr. Kelly Cheshire could not be with us today. But nevertheless, we've got 4 other good panel members. So first I have Savino Segura, who is the founder of Digamma Consulting, who was the laboratory director of Steep Hill Lab from 2011 to 2013 and DB Labs from 2014 to 2016. He worked with representatives from the Washington State Liquor Control Board to draft state legislation regarding cannabis quality control. And he has given lectures on cannabinoid biochemistry and pharmacodynamics, including at the American Chemical Society meetings, as well as CanMeb. Secondly, we have Dr. Daniel Biggerstaff, who is the Technical Director for LGC Standards Center of Excellence for Organic Solutions in Charleston, South Carolina. Dan has over 25 years experience in reference material manufacturing and research, and his current responsibilities include specialized product production, problem solving, new product and method development, training, and customer support. We have Dr. Matthew Wetton with us today, who is the Head of Technical Operations for LGC AXIO Proficiency Testing, and he has over 15 years experience in the field. In his role, Matthew is responsible for the production, development, and technical operation of over 50 proficiency testing schemes, covering chemistry, clinical analysis, microbiology testing, in a diverse range of analytical fields. And prior to joining LGC, Matthew carried out a variety of roles in the fields of phytochemistry and analytical services. And then we have Valerie Bardin, who is the Global Product Manager at LGC AXIO Proficiency Testing. She holds a master's degree in biology from the University of Lyon and has more than 20 years of experience in microbiology diagnostics. She's passionate about the development of proficiency testing, or the development of the proficiency testing range to best meet the constantly evolving requirements of laboratories. So, excellent panel. And we'll try and get through as many questions as we can. If you do have further questions, please add them to the question box. But that's enough for me. So let's get on with the first question to the panel. And the first question is a question we get asked a lot about, which is around the topic of uncertainty of measurement. So the question is, how do you explain uncertainty of measurement to formulators who are looking for one exact number? What would you suggest to formulators when interpreting the laboratory results, and how do you explain that results above 100% are valid? Maybe I could turn to you, Dan, first for that one.
Sure, and thank you, Brian, for the opportunity to be here. It is important to understand that every measurement has some uncertainty associated with it, and that the uncertainty carries forward in each consecutive step in a measurement process. So let's consider broadly an analytical result reported on a sample from a laboratory. The first source of uncertainty is in the reported concentration of the reference material used to calibrate the instrument. In each step in that manufacturing, storage, and shipping introduces some amount of uncertainty. So each weight and dilution step introduces an error associated with the measurement equipment, like your balances and your volumetric flasks. The environmental condition variability in the lab, like temperature and humidity, and also human error, how accurately and precisely is each step performed? Then, how homogeneous is that solution prior to packaging, and does packaging introduce any inhomogeneity? When compounds are mixed together, chemistry happens. The concentrations are always changing. So, how stable is that solution? At what rate do the concentrations change, and how long is the shelf life? Then, what happens when the product is shipped? How do temperature changes affect the concentration? It's the responsibility of the reference material provider to thoroughly evaluate and document these uncertainties and report them as the total combined uncertainty on the certified concentration. It's reported at the 95% confidence level with a coverage factor of 2 for an ISO 17034 certified reference material. If a laboratory chooses to use a reference material that are not CRMs, then they don't even know what the initial uncertainty of their measurement process is. The next step to the uncertainty in that final result is associated with any dilutions needed to prepare the working standards. In most cases, the purchased reference material needs to be diluted to be used on the analytical instrument. This step introduces many of the same uncertainties associated with the initial manufacturing. Then there's the uncertainty of the analytical measurement itself from the equipment. So if you take the same sample and analyze it multiple times, the calculated result will be different each time. So how large is this variability? Typically, in an analytical measurement process, statistical process control, or SPC, is used to determine this variability and make sure that the measurement process is in control. It looks at the average and standard deviation of multiple measurements. In a truly random variability on a measurement that is in control, 1/2 of the measurements will be above 100% recovery. This degree of uncertainty is incorporated into the reported result. Now for the sample preparation. How homogeneous is the portion of the sample used for the analysis? How much variability is in the sample prep method itself? Does the sample prep step introduce any bias? Good testing laboratories will use quality control techniques such as fortified blanks or laboratory control samples to evaluate this and set acceptance criteria, again, based off of statistical process control. Finally, you've got the actual prepared sample that is run. What kind of interferences does the sample matrix have on the reported results? These can be evaluated using matrix spikes, matrix certified reference materials, and performance evaluation testing samples, or PTs. All of these areas of variability add to the uncertainty to the reported analytical result, and it really emphasizes the importance of using high qualified accredited laboratories that understand each of these sources of uncertainty and addresses them appropriately. So Matthew, do you have some comments on that?
Yeah, thanks Dan for that really thorough introduction to measurement uncertainty. From a proficiency testing perspective, uncertainty of measurement is also a very important consideration for both participants and the PT provider themselves. In broadest terms, an analytical method which has a large measurement uncertainty relative to the criteria which is used for the participant assessment is unlikely to regularly perform well within proficiency testing. Proficiency testing providers must think about and they must assess the uncertainty of the assigned value as part of the PT round, because the uncertainty of the assigned value includes contributions from the uncertainty of the characterization of the PT material, uncertainty due to inhomogeneity, uncertainty due to instability of the PT test material, and the uncertainty due to transport. In the event that the uncertainty of the assigned value, so this total uncertainty, is large relative to the assessment criterion, the PT provider cannot just provide assessments without some kind of explanation or caveat, or without including the uncertainty of the assigned value within the calculation of the performance score. The assessment of a participant's estimate of their measurement uncertainty can be included as part of the proficiency testing scheme. However, I would say at this moment in time, that's not commonplace. Savino, did you have any other thoughts on measurement uncertainty?
Yes, thank you, Matthew. So I always explain measurement uncertainty to people who are new to the concept, especially in the laboratory, as the sum of all possible errors that can be introduced from each measurement that happens in the process of sample preparation and analysis. And this includes volumetric measurements like graduated cylinders, pipettes, flasks, and mass measurements such as scales and balances. Take a graduated cylinder for an example. Each time it's used to measure some methanol, you visually line up the bottom of the meniscus with the graduation mark. The final measured volume will vary by a very small percentage with repeated measurements or between different technicians. The measurement uncertainty is a calculated estimate of this small variance. Individually, they appear to have a negligible effect on the final laboratory result, but in assays with multiple steps, they quickly add up. And each piece of equipment that's calibrated by a third party will usually have a calculated uncertainty measurement that is included with its corresponding calibration certificate. A laboratory can also calculate the measurement in-house on its own. They'll follow a standard procedure that's established by a certain body, whether it be ISO, NIST, or other. And the measurement uncertainties from each step in the assay are combined, again, using a standardized equation, and that will produce a final uncertainty of measurement for the entire assay. And this should be reported along with the laboratory results such that the results represent a range of values and put it in context. And then we shoot back to question.
Okay, thank you very much, and I hope that's answered that question fairly thoroughly. One of the things touched upon there in terms of uncertainty and the various factors that we need to look at included to look at any instability that might be present in the sample. And that really much leads us on to another question we've had, which is really around the stability of cannabinoids. The question is, what is decarboxylation and how does it affect cannabinoids? And how much of a role do you believe degraded reference standards and low response calibrations have played in the inflation of potency results? Maybe I would to stand with you, Savino. You could take that one first.
Yes, thank you. So decarboxylation is the loss of a carboxyl group from an organic carboxylic acid. And this usually escapes the molecule as gaseous carbon dioxide, CO2. The cannabis plant itself primarily produces cannabinoid acids, such as THC acid, CBD acid. And these compounds have very little psychoactivity on their own, but still some anti-inflammatory properties. But when the plant is exposed to heat, and this is most greatly done during smoking, extraction, some kind of process such as that, these cannabinoid acids will degrade into their what they call their neutral bases, THC and CBD, using this example. And these compounds are the ones that usually have psychoactivity and usually the ones that are more targeted by these analyses. Now, getting into standards, calibration standards for these cannabinoid acids are very unstable at room temperature, and they will spontaneously degrade into their neutral forms very readily. And so they have to be kept very low as possible. It's usually set around -70 Celsius, but it's basically as low as you can go without the solvent itself freezing. And this includes during shipment. And so the shipment has to be done on dry ice. And so if calibration is performed using a degraded standard, a degraded THCA standard, the THCA peak will skew lower, causing the concentrations of any products measured against that calibration curve to appear higher by comparison. So for example, if the chromatogram of cannabis flower measures there's a calibration created using a standard that was degraded by 20%. That flower will be measured 20% higher in concentration. So say if the flower would normally be measured at having 10% THC acid, it will now appear to have 12% THC acid by weight, which is incorrect. And so to prevent this type of error, the calibration standards have to be kept in sub-zero conditions and spend as little time as possible out of the freezer. And this includes during sample handling for creating the calibration curve itself. And all this stuff has to be done very quickly by the lab technician. And so for the PTs to be properly representative, they've got to be as homogeneous as possible before shipment, and also the shipment times have to vary as little as possible between the different laboratories to ensure that the same, whether or not it's degrading or not, the same amount of degradation is happening for everybody's sample. And I believe Dan then has something to add.
Sure, and Savino described the process very well, and for us, the stability is an incredibly important aspect. And we have put a great deal of research into identifying various stabilizers that we can add to the acid compounds to significantly improve the stability and therefore the accuracy of the analytical results. One thing that we found is that by adding 10% water with a little ammonia significantly improves the stability of the acid compounds. And this makes sense from a chemistry perspective in that what we're doing is we're converting the free acid to the ionized form, and this makes decarboxylation much more difficult. And it really has no impact on the chromatography since the mobile phase already contains water.
Okay, thank you. Maybe just moving direction a little bit. I mentioned at the start that obviously the cannabis has been legalized around the world in the last 10 years, although admittedly legalized in different parts of the world at different times, and still obviously not legalized in some areas. And so we have a question here around regulation. How are cannabis labs regulated, and how does this vary by state jurisdiction? And what is the best way to ensure we are up to date? Valerie, maybe I could turn to you first on that one.
Yes, thank you, Brian. Well, I can start with the European Union situation where the laws on cannabis are very, very diverse. I think we can practically say that there are as many regulations as there are states in the European Union, more or less. So if we first look at medical cannabis, it is authorized in 21 out of the 27 states of the EU. For instance, it's authorized in Belgium, in Italy, in Spain. In France, it is under a 2-year experiment, 2-year at least probably. And in the UK, which is not part of the European Union, part of Europe, medical cannabis is legal. When it comes to recreational cannabis, there is a lot of talk about possible decriminalization or even legalization of the product, but in the end, the possession of cannabis for recreational purpose is prohibited in all member states except one, which is Malta. Well, that said, we know that there are some countries that are quite tolerant about the possession of small quantities of cannabis. And it's likely that the 2 countries that are next to legalize the possession of cannabis are Luxembourg and Germany, but it's a process that takes time. So with so many different regulations, there is obviously no European directive related to the quality of medical cannabis. So most labs that are testing cannabis will test for cannabinoids and terpenes, which are the most popular analysis, but also chemical and microbial contaminants. And for instance, aspergillus and the mycotoxin risk that is associated with the presence of aspergillus is especially scrutinized in I don't know if the situation is quite different in the US. Probably a question for you.
Savino, perhaps you'd like to.
Yes. So the laboratory testing standards for cannabis in the US are pretty much the same as in Europe, as you were describing, where it varies state by state. And the regulations are set up in each state's individual law as it has deregulate or legalized cannabis. These lab standards happen in that legislation, and they require specific analytes, such as specific cannabinoids, sometimes terpenes, and specific contaminants, pesticides, solvents, metals, microbes. And they also dictate the action levels and pass-fail criteria, and as well, the permitted variation in reporting, the certified reference materials and PTs. And while this is typically a multi-step process that includes some public input at some point, large states like New York and California don't take much stock in this public input. And the reason this can be a problem is that many of the details of the law are usually written by large lobbying groups and special interest groups that don't know much about the underlying science or the practical implications of current testing technology. And the lawmakers know even less. This can result in requiring extremely low quantification limits for some compounds. And some of these limits may not even be possible in complex matrices such as the cannabis plant, using even the best technologies of the industry. So this often requires adjustments to the regulations once the state reference labs attempt to and fail to achieve these set reporting limits. And so the best thing to do is to follow each state's regulatory body and track the changes as they're happening. And this obviously is difficult because there are 50 states, and so you have to follow 50 different legislative bodies, but unfortunately that's the way to do it right now.
Okay, thank you. Maybe we could just turn our attention a little bit around testing methodology, and one of the key areas of testing that perhaps gets talked about quite a bit is in terms of potency. And the question is, there is a good deal of market pressure on potency testing methodology. Is there any comment from the panel on the efficacy of different methods? Maybe I'll stay with you, Savino, just to kick this one off.
So in terms of methodology, assuming all the SOPs are followed as written, the biggest factors influencing accuracy in the analysis are sample preparation and the chromatographic separation. The samples have to be selected from the batch in a randomized and representative manner. The material must be sufficiently ground and homogenized for optimal extraction, utilizing all parts of the submitted plant matter. Some labs have attempted to skew towards higher cannabinoid results by omitting parts such as stems and seeds, which add mass but are relatively low in cannabinoid content. And the chromatographic method, including the choice of column, mobile phases, and gradient, has to sufficiently separate the major cannabinoids, not just from each other, but also from the minor cannabinoids that are also in there. And the methods that attempt to sacrifice this resolution to reduce runtime, they end up lumping some of these minor cannabinoids in with the peaks of larger cannabinoids. They co-elute. And this makes these major cannabinoids appear larger, the peaks appear larger, and the final concentration will appear greater. And in my experience, if the method is able to separate the main 11 cannabinoids that are most common to baseline, then that method is probably sufficient enough to separate the peak areas and sufficiently pass accurate PTs. Now I'm going to turn it over to Dan, who's got some other info.
Sure, and Savino is absolutely correct on the method needing to achieve good resolution and the problem of coelution. The most common analytical detection system for cannabinoid analysis is the UV absorbance using a diode array detector. The technique has really good precision, but relies on separation and retention time for identifying a particular cannabinoid. Because the detector is not selective, any matrix interferences with the same retention time that absorb at the measured UV wavelength will increase the area of the peak and thus bias the result. Some laboratories, to avoid this, will use HPLC mass spectrometry to analyze the cannabinoids. Mass spectrometry is a very highly selective technique and can minimize coelution problems, but it's also less precise. Matt, do you have anything to add?
Thanks, Dan. Lots of really good information on why methods may not give the same result if they're not running correctly. From the point of view of proficiency testing, PT can be a powerful means of evaluating different analytical methodologies. Where there's a sufficiently large data set and a large number of participants within the PT, the method information that we obtain as part of the reporting process can enable the comparison of performance of methods based on individual methods or on groups of methods which are technically equivalent. So when we look at this data, the methods which perform more or less well than other methods can easily be identified in terms of accuracy and precision over a range of different matrix types, over a range of over a period of time, and over a range of measure and concentrations.
Okay, thank you for that. And obviously potency testing is just one area of testing that that the cannabis testing labs need to be concerned with. Another area is microbial test methods. And a question here in terms of microbial testing is, what are the advantages and disadvantages of using molecular techniques, such as PCR, and traditional techniques, such as culture plating? Valerie, let me turn to you on that one.
Yeah, well, when we consider different methods or techniques, there are of course always pros and cons for both, but PCR methods are, generally speaking, faster, not to say much faster, compared to traditional or plating methods. With the PCR methods, the labs can get results within generally 1 or 2 days, when it would take up to 5, 6, 7 days with culture methods. So, the time to get a result is of course an important factor. PCR methods are very well adapted when it comes to pathogen detection, because they will give a presence-absence result, and this is what regulation is for pathogens. So it's interesting to see that a good number of molecular methods are validated by the AOAC for the detection of pathogens in a cannabis matrix. So today there are 7 methods that are AOAC validated for the detection of Salmonella, 8 for STEC, and 4 for Aspergillus. So those ones are really the main pathogens when it comes to cannabis. So they are all PCR methods, and as far as I know, there is no cultural method, so plating method, that is AOAC validated for the detection of pathogen in a cannabis matrix. There are also a handful of methods that are AOAC validated for yeast and mold enumeration. Most of those methods are either cultural or MPN, the most probable number methods, and I think that is only one that is molecular based. So really, it's very much PCR when it comes to presence-absence, and more traditional method, well, non-PCR based methods when it comes to enumeration. If we look at Europe, the situation is quite different, because there is just no method at all, neither cultural nor PCR, that is validated for detection or enumeration in a cannabis matrix. This may change in the future, but today we don't have any validated method in Europe for cannabis testing. Savino, you may have some thoughts on this PCR versus cultural question as well.
Yes, thank you, Valerie. So the molecular techniques allow much better speciation, which has been in line with how most current state regulations are. They're moving away from total yeast and mold count, which is what they used to do. They used to rely everything on counts such as that, on plate counts. And now they're moving towards measuring only certain pathogenic strains. Now, these molecular techniques cost a great deal more than plating. The cost, used to be plating was one of the cheapest ones you could have in a laboratory, and now the molecular is one of the more expensive. But they provide faster results than plating. And this has become a major point of competition between laboratories because here in the US, as it's all private industry, clients do not want to wait very long for the results, and they'll tend to go with laboratories that can provide them with the fastest results, even if it's just a difference of 12 hours. And that's whether or not it's practical in the given situation. But the quantitative PCR is relatively new compared to counts that are done from plating. And so the standard methodology is only just starting to catch up, as Valerie mentioned, that there aren't as many standardized methods in general, and especially for cannabis, standardized methodology is very new. And I'll shoot it back to Brian.
Okay, thank you. And just like Matthew mentioned before, even in microbial sort of testing, proficiency testing plays an important role in enabling laboratories to compare performance between the different types of methods. And that perhaps leads us on to the next question, which is specifically on proficiency testing. And it says, can you explain the importance of participating in a proficiency testing scheme? If labs can be 40% above and below a target, are we operating at the same standard? Let me turn to Matthew for this one.
Thanks, Brian. Well, in terms of the importance of taking part in PT, proficiency testing is one of the most important tools in a laboratory's QC armory, and it's a truly independent method and probably the only form of quality control where a laboratory doesn't know the answer before they start the testing. So that's an important point to make for the start. When we think about the assessment criterion for a given proficiency testing scheme, it's tied to the aims of the scheme itself and also to the testing labs who are taking part. In most cases, it's also a bit of a consensus and may not suit every lab completely. But when we think about these particularly large uncertain or these particularly large criterion, it may be that the PT scheme is targeting a specific type of testing lab or a specific methodology. If in these cases the measurement uncertainty of the method which is being tested is small, then actually I guess what the customer is saying is correct, that a performance criterion of 40% may not be suitable, because if the labs are much better than that criterion, they don't face a realistic challenge, and they'd be unlikely, as the PT progresses, to receive anything other than a satisfactory performance score. If, however, the uncertainty of measurement is larger, then actually a performance criterion of 40% may well be entirely appropriate. A good example of this would be when you're considering concentrations of measurand which are very low in something like the microgram per kilogram range. And as the relative spread of the PT data increases as the concentration gets lower and lower, then actually a performance criterion of 40% may be actually sensible given the performance of the participants.
Thank you.
Davina, do you have anything to add to that? Sorry, Brian.
It's okay.
Yes. So as mentioned previously, the quantification of low-level contaminants can be very difficult in a matrix as complex as the cannabis plant, especially when taking into account having to do quality control matrix spikes that have to come back within a certain recovery in order to pass. The analytes most prone to this degradation can also vary greatly in concentration from manufacturer to manufacturer of both PTs and of calibration standards. And so a good point of action for a PT provider would be to inquire about the specific lot and manufacturer of the calibration standards used by the lab in deriving the results that they submit for the PT. As well, internal standards are whether done by the PT or added later by the participating laboratory can be very important because if not performing a daily calibration and a matrix match calibration, these internal standards become necessary for these very, very low level contaminants. And as well, having a standard blank matrix for spiking. This is something that's really important that exists with a lot of other industries, with a lot of other types of matrix, where there is a standard matrix that can be purchased that can be then spiked with a variety of different analytes. And so what we need for cannabis is to have a blank matrix that's standard, that can be spiked and can also be spiked for different analyses, and not this one can just be spiked for metals, or this one can just be spiked for pesticides, but it's not free of metals. We need one that's free of all contamination and can be spiked for any of them. And my last point is, as we mentioned earlier, is to have the measurement uncertainty calculation from the laboratory go along with their submission to the proficiency testing manufacturer. Shoot it back to Brian.
Okay, thank you, Savino. And maybe I'll probably stay with you for the next question to start with. But anyway, the question is, how do terpenes and cannabinoid combinations impact the overall effects of cannabis? And how does this impact the testing and analysis of the sample? So as I say, maybe stay with you, Savino, firstly.
Okay. So although the objective research is scarce on this topic, there's very strong anecdotal evidence that the perceived differences in reported effects between different strains of cannabis are caused primarily by their terpenoid profile. We have the monoterpenes and sesquiterpenes and some of their corresponding alcohols and oxides. They created the distinctive smells and flavors of different cultivated cannabis strains. And now between these strains, all these terpenes are typically found, but will vary in their relative concentrations between different strains or cultivars. We call them strains in cannabis. The cannabinoid profiles, on the other hand, are usually pretty uniform and tend to occur in three main clusters: THCA dominant, CBDA dominant, and then split, where it can be a 1-to-1 to 1-to-3 kind of ratio between THCA and CBDA. Now, probably 90% of the flowers fall into one of those three clusters. Terpenes, however, they'll create many more clusters. And we've detailed that, and you could reach out if you have any other questions on our past research, but we've detailed a lot of the different clusters of terpenes that can occur in these plants. And right now, only about 20 to 50 of the over 400 known terpenes are tested regularly in laboratories. And this is mostly due to cost. There's a high cost of quantifying these terpenes, and there's a very low return in monetary value of quantifying them. So many times organizations will only test them either if they have to, or if they want to advertise that their strain has some particular terpene profile that makes it unique or desirable. All right, Kelly? Oh, sorry, Dan.
Dan.
There is a growing interest in the terpenes, and it's not only from a scientific perspective, but also from a commercial perspective. You know, on one hand, as noted by Savino, terpenes are responsible for the distinctive flavors and aromas associated with different cannabis strains. And these sensory factors feed into the consumer's enjoyment of a product. So consequently, it could be beneficial for companies to create custom terpene profiles to target different audiences and possibly improve their sales. You know, on the other hand, you also have what is commonly referred to as the entourage effect, where combinations of certain terpene and cannabinoid profiles give rise to different therapeutic benefits for things like anxiety, sleep aids, pain relief, autism, and seizures. The key in both situations is for a producer to be able to consistently reproduce these profiles between batches to ensure that that same desired sensory or therapeutic effect is achieved. So for testing, this might mean increasing the panel of analytes that you're looking at to both qualify and quantify. This also means it's important to have robust SOPs in place with well-documented method validation using high-quality reference materials and regular participation in PT schemes to demonstrate the competency of the laboratory to both auditors and customers that the profiles that they're generating are accurate. Also, Israel and Thailand are actively doing research on medical applications of cannabis, and a somewhat common finding is that one observed therapeutic benefit are not duplicated when isolated cannabinoids are tested, leading to the idea that that therapeutic benefit is the result of multiple components in the cannabis plant. Again, the entourage effect. It has also been observed that different strains do not always produce the same therapeutic benefit. So as this type of research matures, the testing will develop to quantify the therapeutically relevant components. And back to you, Brian.
Thank you, Dan. I'm interested in a minute to talk to the panel about perhaps trends and future trends, but it's just a couple of questions I might just pick up that's come from the audience, which hopefully are quite quick. And one perhaps for you, Savino, is it says, I know that cannabinoid standards have to be kept frozen between use. Is it necessary to store the standards at -70 degrees centigrade?
So the cannabinoid acids, those standards will do best. Now, I personally usually stored them at -20 once I cracked the vial and started using them, because I usually would go through them in a matter of 2 weeks. But if it's something you're storing for longer than that and it's still coming from directly from the manufacturer, I would say store it as cold as possible without freezing the solvent that it's in. The cannabinoid bases will usually do fine in a standard freezer for up to a few weeks, definitely, probably even a few months, based on my personal experience. But yeah, I would say when possible, store them as cold as you can.
Okay, no, thank you, Savino. And a quick question for you, Matthew, is does the proficiency testing organised by LGC fulfil the requirements of ISO 17043?
Yeah, thanks, Brian. At this point, because the proficiency testing is in a trial phase as we bring more matrices online, the sample isn't accredited to 17043 at this time. But like all of the PT test materials produced by LGC, whether or not they are accredited, they're all produced using the same processes and to the guidelines of 17043.
Okay, thank you for that. So let me now actually turn to the future. I mean, obviously, I've already said it's a fast-moving field. And a question we have here is, looking into the future of cannabis testing, how can you support us in this rapidly expanding landscape? So it'd be good to hear from each of our panel members on this one. Let me start with you, Dan.
Sure. One thing we can do to support the industry is in the development of stable reference materials, especially the matrix reference materials. Product matrices are already fairly complex. For example, we've got gummies, got chocolate, oils, just to name a few. So as products evolve, having RMs as close in composition to the testing material or the material being tested as possible would go a long way in having more realistic analyses and conclusions as to the validity of those. I also suspect that as the cannabis industry matures, contaminant testing, especially like pesticides, will evolve from a targeted list like it currently is to a more comprehensive multi-residue screening list to identify the presence of more unapproved pesticides, very similar to how the food safety industry testing has evolved. Savino, your thoughts?
Thank you. First thing is to look into the sample prep methods for these different matrices, especially new ones, patches, more complex ones like gummies, chocolates, and any other new forms of delivery that are popping up, and make sure that the PT manufacturers themselves are using the best methodologies in-house to make sure that their results are as accurate as possible. And another one is testing the stability of molecules that are particularly sensitive to degradation, such as cannabinoid acid and some pesticides, testing their stability in different matrix types to make sure we really get the proper pass-fail criteria for each test. Also testing the manufacturer's own in-house calibration standards and stock solutions against other popular market options to anticipate the participant results. If we see the spread of these different possible calibration standards from different manufacturers, it may sell even more, LGC calibration standards, say, if we find that those are the best and most consistent. And finally, sharing these results with lawmakers. These lawmakers are usually pretty clueless and have special interest groups being fed in their ear when they're writing this legislation. And so sharing these results with lawmakers, and this can help establish proper quality control standards in every single jurisdiction. Here in the US, every state kind of has the ability to set their own quality control criteria, and having this information from PT manufacturer would actually give them a much better idea of what the limits and possibilities are of our current technology. Valerie, what do you think?
Yeah, and that's this idea of sharing more with lawmakers is definitely an interesting point that we should explore more. Well, when it comes to PT, the future of PT is, as what said just before, is to better answer the customer requirements, and the labs are really seeking to real matrices as much as possible. This is always a challenge when we are talking about a cannabis matrix. So yes, of course, there are some real logistical challenges, but just I would like to mention that we are today working to provide in our PT scheme, in our cannabis PT scheme, a real cannabis matrix. This was not the case previously because we were just providing a hemp matrix. But this has been a request from laboratories to work with a real cannabis matrix, and this is something that we are now doing, and we will have our first PT round with this real cannabis matrix during the spring. So we will communicate more about this very soon. If we look further, I will just confirm what Dan and Savino have just said. So next step will be to develop PTs with different matrices. This is a clear demand from laboratories, and I would say especially from the custom laboratories, because they face so many different matrices, whether it's edible, beverages, cosmetics, resins, liquid for e-cigarettes, etc. And they are always looking to matrices that really mimic what they have to test in real life. Matthew, you may add something more technical to add to this.
Thanks, Valerie. And I have to say I completely agree. And it's clear that as part of the development of cannabis and cannabinoid testing, PT does have a role to play in the support of these new analytical methods and in the analysis of new and novel matrices. from a PT perspective, it is most useful when the test materials which we're using for proficiency testing closely resemble or where possible are identical to the samples which are routinely analysed by the participants in their day-to-day work. That's the best way for them to be reassured that their methods are working correctly. So, you know, new PT product development in the areas which Savino has highlighted are going to be key in ensuring that PT participants get the most reliable assessment of their performance.
Okay, thank you, and thank you for sharing your views for the future. Interesting, Valerie, you talked about potentially to develop PTs in new matrices. We did have a question. Sorry, I just to find it now. Well, anyway, there was a question asking whether there was any future in terms of developing a sort of a matrix in T, for instance, for cannabis testing. So I think you've probably answered that anyway.
Yeah, that's interesting. I can't remember that T was on the very top of our list because this is not a request that I don't think this is a request that we ever had. But well, we are open to any development. It's all about, well, it's always with PT. It's all about the potential number of participants will be interested in this matrix, because I wouldn't say that basically we can provide anything, but we can provide many things. But if we always need to meet a sufficient number of participants so that our statistic group is large enough to have some decent statistics.
Another question I do have here is, what is the best way to prepare cannabis samples to get the best repeatability on GC and HPLC? I'm probably going to look at either Dan or Savino. I don't know which one of you would like to take that one.
I'll jump in.
I would pass that to Savino.
So I'd say there's no one best way, but I touched on some of it earlier, whereby freezing and grinding the plant sample as much as possible is the best thing one can do, making sure that no fine particles stick to the side of your container, because those are probably going to have trichomes in them. Other than that, simple weighing and then extraction in whatever extraction solvent you choose. My personal favorite is methanol, because it's very effective and low cost. And then you could follow up with vortexing and or sonication. Those are techniques that I like to use a lot of times in concert with each other to make sure that I get the most amount of extraction from the plant sample. And then actually just simple filtering and injection is good. And now with, you know, this tends to work for GC and LC. And actually a little story, when I first started in this industry 12 years ago, we used to use hexane for the extraction on the GC because it would leave behind a lot of the chlorophylls that would tend to clog up the instrument. But it's very difficult to obtain and then dispose of hexane. So we've moved to methanol mostly and moved to HPLC.
Okay, thank you, Savino. I think we've probably got time for just one more question, and that's an important question coming in. It says, can LGC support with import licenses for RMs and PT? Obviously, we're talking about in the world of cannabis. Dan, do you have any comment on that?
Yeah, we actually have a good compliance group that does keep up with import-export regulations and making sure that we do comply with all government compliance regulations, whether we're importing into the U.S. or we're exporting out of the U.S. So I would answer encouragingly. I don't know personally all of the regulations, so I don't want to say yes, we can ship anything and do anything. But Our compliance group is incredibly knowledgeable, and we have compliance people in both the U.S. and the EU that are very knowledgeable and up to date with the various import-export regulations.
Okay, thank you. Valerie, was there anything you wanted to add? But I assume it's pretty much on the same lines.
Yeah, no, I absolutely agree with Dan. We have a fantastic compliance team who are doing all their best to help. And there are so many different situations that, we have to take them one by one, because I wouldn't say that each case is different, but we know that there are some countries where it's quite easy to ship, some where it's quite uneasy to ship, and well, we just take them as they come, and we help where we can. That's it.
Okay, well, thank you. And I don't think we've really got time for any more questions. I know that a number of you have sent through questions, and we will certainly address those questions. So we will answer those by email. So I apologise that we have not had a chance to get around to all the questions. To keep up to date with reference materials and new PTs, please always keep an eye on the LGC Standards website. But apart from that, it leaves me to thank all of you, the audience, for listening, and I hope you found this useful. And a big thank you to me for my 4 experts today who have answered your questions. So thank you very much, and bye-bye.
Thank you.
Thank you. Bye.